Hello, everybody. Put in presentation mode. So thank you very much for inviting us here to be today. So my talk, I wanted to present to you the principle of manufacturing of bacteriophages and some common practices we have for the production. So JAFRAL is a company. We are world biggest manufacturer of phages. So we produce bacteriophages under GMP standard that is used for human therapeutics or veterinary purposes as a drug. And we produce also phages that are used for other applications such as food industry, cosmetics, agricultural application. So we have a wide variety of on which fields the phages can be used. So for these purposes, every field in which the phage
Sorry,
is
Dr.
used...
Hubad, to interrupt.
Yes.
Could you please drag the Meet Google that's popping out on your screen? So could you please drag them down so that we can see the whole slides? I know, stop sharing. Just just drag them down. Yeah, drag them down, please.
Okay, like that?
Yeah, thank you very
much.
Oh
my
yes,
God, I'm
thanks.
so sorry. Okay.
No, that's all right. Thanks.
Okay, so for each application, we have a different certification that allow us to produce that and provide to our clients. For the human therapeutics, we have a GMP certificate that is used for, is a standard for the production under a good manufacturing practice, and it is used for human therapeutics and veterinary purposes. So at least in Europe, because we are based in Europe, phages are classified under biologics. If they are native phages, or if they are genetically modified phages, they can also be classified under advanced therapy medicinal products. These are details, but maybe it is of your interest just to know that there are different classifications also for phages can be applied. Then for food supplements, there are some food regulations for cosmetics, for agricultures. So this all needs to be kept in mind if you want to use those kind of phages in different applications. Usually what we use is that phages are classified in biosafety level 1, some phages also 2, and we can use non-GMO or GMO phages that can be produced, and they were already used on the field. So okay, so maybe just a little background. We have quite a lot of track record for the production of phages, and specifically for the human therapeutics, we produced already 14 drug products that were successfully used in the clinical trials. We're also involved, at least in Europe, the regulation is for the phage field is quite evolving, and there's an EMA guideline now that was available for draft comments for the human therapeutic. Also, EMA has the guideline for veterinary, and also there is an European Pharmacopoeia. There is a general chapter on the preparation of phage products, and we were all involved in all those preparations of all those drafts and comments to these activities. Okay, so maybe because we are a CDMO, that is a contract manufacturing development organization, we can provide you with a list of, for example, what is the most wanted phages that we get order from. So usually the most requests come for Escherichia coli, for Pseudomonas, Staphylococcus. These are the 3 most common phages that are currently produced, but also Klebsiella is quite popular in the sense that there are quite a lot of infection for the phages and so on. So at least up to now, whatever we got a request, we were able to find phages either from the nature or from some other environment. So how does the development of the phage product look like? So it all starts with the bacterial infection, if we're using phages for the treatment. So we can help with the phage selection from the environment. We do the phage hunt or phage design. Some clients also already have their own phages. In that case, we start with the process development. So to successfully produce phages, we need to work on the successful process development that will produce phages with high yields in desired purity. And in parallel, we need to ensure that the produced phages will be stable. So usually we start with small-scale process development, and then put this on stability studies so that we know that also those phages are okay. Okay, and then if a larger scale is needed, once we do the scale up, we can either move to GMP production or to other kind of production, which we call this non-GMP, but usually this is for other applications that are not human therapeutics. And what is the most important in the phage development of the phage product is to ensure the stability of the product in some kind of relative long term that can be then used. So in parallel to all the process development activities, we need to do all the analytical developments of phage product that will then support the process and will support the stability studies. So usually we do initial development once we have the host and once we have the phage. And then if we move to production that will be used on the field, we do some kind of method qualification so that we know that the method is suitable for its intended use. If it goes for human therapeutics in clinical trials, we also need to do method validation because this is regulated. And then after all these activities are done, the method is developed and it's shown that it's suitable for its use, it can be used in the release testing of the produced product so that everything is controlled from the start to the end of the process. So the quality control phage-based product pattern, what is used as a release, it really depends on the application that we do, but usually it includes like appearance test, pH, potency to show how many phages are in concentration of phages, morphology, identity, sequencing is relatively a must in the later years because of the advances of the sequencing platforms, endotoxins, microbial purity, like bioburden, sterility, and other impurities that arise from the bacterial host that is used in production. And also, as I said before, after everything is produced and it's ready to be used, we need to show that if the phages, and not everything will be used at the same time, so we need to show that stability will be maintained until the product is used. Sometimes this can be after 1 year, sometimes this could be after 2 years. Usually we aim that the produced phages are stable at least 2 years. It is true that the majority is stored at 2 to 8, but the stability conditions really depends on the phage itself as well as on the formulation that is in which the phages are stored. So in addition to process development and in addition to analytical development, one important aspect in the production process is also the final formulation. And there are a lot of formulation studies that needs to be done also in parallel to show that these excipients that are used in the final buffer that are suitable and compatible with the phage and that they enable phages to maintain its potency throughout the shelf life. And to determine a shelf life, also stability studies are done, but these are usually in parallel to the already produced material at the different temperatures so that we can also extrapolate maybe if the phages are stored at 2 to 8, and then we can put them on accelerated conditions just to show that to accelerate its aging, and we can maybe anticipate that shelf life for them for long-term stability at 2 to 8. So, and then how the final product looks like the majority of products we produce are in liquid formulation. They can be in small vials that can be used for injectables or any other application, or they could be larger bottles that are then later processed or either diluted on the field or anything else. And also we have a dry powder option if liquid is not desirable. Usually the dry powder would prolong the shelf life. However, there are some losses already during the production, during the drying, so it needs to be seen what the best option for the client is. So that is like a brief overview of how the production process looks like. If you have any questions, you can also contact us on the email that is written here, or maybe at the end of the session with the Q&A session. I'm also very happy to reply to your questions.
Thank you so much,
Thank you so
Dr.
much.
Hubad. That's really insightful, and it seems like it's a dream to see like how phages are evolving from the lab to the markets, and really interesting. Even I have a couple of questions which I would be asking. Really impressive presentation. Thank you so much.
Not a problem.